Tonight I want to ask the question, can life make itself from nothing? Life is a very special entity. You can think of life as dead chemicals that have been pushed up to an energetic level where their level of entropy is sustainable, where their level of integration one with another is so compact that essentially we can't see a single cell, most single cells, with the naked eye. So life is like pushing a rock up a hill. And you could, as long as you push the rock and expend energy in that process, the rock will stay up on the slope going up towards the top. But if life should expire, everything goes back the other way, and entropy takes back over. And the question is, how did life get to be the way it is? Can it be made from nothing and from dead materials? That's the question. And in order to do that, I want to take two case studies. The first case is an object that I found recently in my backyard. It's a strange-looking object. It's a large stationary object. Looks a little bit like some scaffolding or a skeleton. It appears to have a weathered texture, a little bit rounded on some of the edges. And so I wondered, where did this object come from? So I did a structural analysis, and there are 4 points of contact with the ground. And it appears as if any weight, its own weight, would be transferred through its members onto the ground. Doesn't seem to have any movement at the present time. And a stress analysis suggested that this is a designed feature. And although we know that, of course, this object isn't designed, it must have made itself. And so, like any good scientist, I reverse engineered it. I took it apart, as we do with life. We start with a completed structure in life, and we take it apart to see how it works. And that's what I did. I noticed that as I set the parts up, that it has bilateral symmetry. Interesting kind of a body shape or a plan in several of its structures. In fact, 4 different components have, maybe 5 have bilateral symmetry. It also has these interesting piercings all the way through many of its members at the ends. Interesting feature that we don't really understand. Holding those piercings together, those parts together, are these strange metal alloy structures. They're not part of the normal structure of the material themselves. And as you look at them, they appear to have a coiled inclined plane design. And on the opposite side, through the material, is a complementary coiled locking ring that allows these structures to, in a vise-like way, pinch in. Some of the fasteners are actually quite long, and they pierce through the material in very, very small circular holes. And in fact, I've placed a coffee bean there next to the hole to give you a sense of size of that hole. And I want you to remember the size of the coffee bean, because the size of the coffee bean is going to be important later on. Now, further analysis, we did C14, carbon-14 dating on this object, since it's made out of carbon. It's less than 5,000 years old. Accuracy wasn't real good on the carbon dating. The major composition, as we did some mass spectrometry on it, was cellulose and lignin. And there you see the structure of cellulose. So it's clearly a carbohydrate kind of a structure. This material we find naturally occurring in great abundance all over the world. Large structures and clearly easily accessible to many, many people. But it's a naturally abundant substance. If you go around, you also find that there are some naturally occurring groupings of this material. It seems to fall in distinct patterns. Some of these patterns are rectangular, some are long spindle shaped, some are straight, where their sides are juxtaposed. And clearly this is a natural feature that just happens because of the interaction of the material with itself. So in thinking about this, and recognizing that this is a structure that must have built itself, I wonder if we could think about a possible scenario of how we could use energy plus time plus raw materials to see how we could understand how this object would come to be. And so clearly in the natural world there's lots of energy. There's wind, there's storms, there's wave energy, and there's lightning energy. And that lightning can have severe effects on the naturally occurring substance. And you see here a sort of a broken section that was created by just such a lightning strike. It appears to match fairly reasonably with what the material is near where I found the original object in its striated pattern. And then from those broken pieces, many of which would have straight edges, and there were some early processing steps. The edges would have become rounded and the ends would have become pierced. Don't quite understand what mechanism would pierce the object like that naturally, but it's under further study. I have to write a grant about that, I think. There's also must have been a selection process to remove defective parts, and clearly you see a defective part here, a large knot and sort of a discolored portion of the wood of this material that ended up not being incorporated into the structure. And then clearly self-assembly would begin. Once those pieces have a shape and then the natural properties of the material take over, and it just begins to normally self-assemble. And so you see the back taking shape, and then the curvature of the back seems to be important as the curvature of the base. And in the final stages, the object comes to its completed form, the form that I found it in my backyard, the final object. So clearly this is a work in progress to try to understand all these steps of self-assembly. But I wondered, going beyond that, what would be the structure that would be around this? This appears to be scaffolding. It could be like a bone structure for some object. And so if you think of this as more of an internal skeleton kind of a structure, I posed this to an artist, and I said, what possibly could this have looked like when it had flesh on it? And he came up with this. And so this, I'm sure, will be published in National Geographic very, very soon. That's the first case, scientific study of that particular object. Now, there's a case study of another object, and this object is quite different from the first case study. This is a very, very small object, orders of magnitude smaller, thousands and thousands of times smaller, millions of times more complex than the first case study. In fact, it requires very specific temperature conditions to even survive. It requires a certain food supply of glucose and fetal bovine serum and other careful conditions of nutrients to keep it going. And you'll notice down in the right-hand corner that there's that bean again, that coffee bean. Because although the picture here is very large, I want you to appreciate actually how small this particular object is as we look at it. So what I'm going to do is ask you to look at this animation of a size and scale comparison with the size of a coffee bean, a grain of rice, and a times 12-point type on a typical word processor. And so you see the size of that coffee bean. And now we're going to explore, as I start this, as we go down deeper in this animation, we'll see first a sesame seed. It's a typical small seed. And a grain of salt, just for reference. Now we begin to get, at 500 micrometers, a size of typical cells. Paramecium, a skin cell. Human egg is actually quite large by comparison to a skin cell. A photoreceptor rod that's found in the eye. Go a little deeper. Of course, you see the sperm cell, much smaller than the egg cell. And then we see a red blood cell, very familiar to all of us, a non-nucleated hemoglobin-containing oxygen transporting cell within our blood system. And then a chromosome, a condensed chromosome, 7 micrometers. And then we start to get down to yeast size, Baker's yeast. And then we find that we're at a size where we need special light magnification at the size of a living bacterial cell in order to be able to see it. And beyond that, at a deeper level, there are structures that would only fit within a living cell, like a lysozyme or a mitochondrion within a eukaryotic cell. So we'll go a little further down, and we start to find viral particles, particles that have a nucleic acid and a self-assembled protein capsid, but are very filterable. They're very difficult to filter out, and they cause various diseases. They're very, very tiny particles. Hepatitis virus, rhinovirus, which is the normal cold virus, and below the level of the smallest of viruses illustrated here, we begin to see subcellular organelles, the real working parts, the little machines within the cell, the ribosome, which translates RNA, a transfer RNA, which brings amino acids up into the ribosome, the hemoglobin molecule itself, which binds oxygen and transfers it to tissues, an antibody, this bifurcated molecule, 12 nanometers in size, that is part of the immune system, phospholipids, which make up the cell membrane. And as we go deeper down in the complexity, we see the water molecule itself, and a glucose molecule, a 6-carbon sugar, an adenine base, part of a nucleotide, a methionine amino acid, part of a protein. And then as we finally focus in, at least on this magnification, we find the carbon atom itself. Life is based on carbon, hydrogen, and oxygen, 340 picometers in diameter. And now just appreciate, as we zoom out slowly, the size and the scale of what's within a living cell. The components, sometimes viruses that take over. Other bacterial infections can certainly be within cells, like mycobacterium. The cells themselves, their genetic components, various what are called molecular machines within the cell. And as we zoom out, we finally begin to see the edge of the grain of rice, or the edge of the coffee bean and the sesame seed, and the 12-point type. And so what I want you to appreciate is that this case study is very different from the size and scope of the first case study. Vastly more complex, vastly smaller, and vastly more difficult to actually tease apart in ways that we can understand. Not only does the complexity make it difficult to tear it apart and understand, but that complexity involves information flow as well as complexity. And so I'd like for you to listen to a little bit of a description of what it's like to go into a cell and then into the nucleus of a cell and see its genetic machinery, its genetic material, its DNA, involved in transporting information from the nucleus out into the cytoplasm, where another molecular machine aids it, the ribosomes, in creating a protein. So let's watch this.
In the years since Kenyon's rejection of chemical evolution, science has revealed the details of an entire system of information processing that bears the hallmarks of intelligent design. With computer animation, we can enter the cell to view this remarkable system at work. After entering the heart of the cell, we see the tightly wound strands of DNA, storehouses for the instructions necessary to build every protein in an organism. In a process known as transcription, a molecular machine first unwinds a section of the DNA helix to expose the genetic instructions needed to assemble a specific protein molecule. Another machine then copies these instructions to form a molecule known as messenger RNA. When transcription is complete, the slender RNA strand carries the genetic information through the nuclear pore complex, the gatekeeper for traffic in and out of the cell nucleus. The messenger RNA strand is directed to a two-part molecular factory called a ribosome. After attaching itself securely, the process of translation begins. Inside the ribosome, a molecular assembly line builds a specifically sequenced chain of amino acids. These amino acids are transported from other parts of the cell and then linked into chains often hundreds of units long. Their sequential arrangement determines the type of protein manufactured. When the chain is finished, it is moved from the ribosome to a barrel-shaped machine that helps fold it into the precise shape critical to its function. After the chain is folded into a protein, it is then released and shepherded by another molecular machine to the exact location where it is needed.
This is absolutely mind-boggling to perceive at this scale of size such a finely tuned apparatus, a device that bears the marks of intelligent design and manufacture. And we have the details of an immensely complex molecular realm of genetic information processing. And it's exactly this new realm of molecular genetics where we see the most compelling evidence of design on the Earth.
That was a short vignette from a DVD titled Unlocking the Mystery of Life by Illustra Media. And I recommend that you pick that up if you can. It's a wonderful, wonderful documentary on complexity and life. In every one of the trillion cells in your body, you have that kind of molecular machinery within the structure of the nucleus expressing hundreds of genes just at the right time. So when I say the complexity pales by comparison to this complexity is staggeringly greater than the first case, I'm only really underestimating. And as I look at that, I want to tell you that we've just touched the surface of the complexity of a single cell. And so as we look at that and we appreciate it, we ask, can life such as that make itself from nothing? And of course, that's the question. Where did life come from? How did it get to be the way it is? And there are two basic opposing beliefs. One is called spontaneous creation, or spontaneous generation, might be the more familiar biological term, and special creation. Those are the two opposing views. We've seen this slide before, where nothing created something. But now the added fact is that that something was dead stuff, dead chemicals, that suddenly somehow became living stuff and then turned into everything. And that happened supposedly by a natural process. The normal interaction of matter and energy, the normal attractive forces of certain chemicals and proteins. By comparison, the special creation theory says no, there's a fixed point, and that fixed point is God. And God created the heavens and the earth. And God created those heavens and earth to house life, just like we've seen, just like we are, as a matter of fact. But that there have been some problems in life since that original creation, and it was cause for judgment, and we are now all subject to decay. But those are the two opposing beliefs. One says on the left, nothing created something, and then it began to improve itself. It became alive, began to improve on its structure, and eventually became aware of itself and created culture, created everything that we are and everything that we know. It's been a story of great improvement. The position on the right says we started out much better than we are currently. We started out in a better state, in a state of perfection, where there were no mutations. And there have been problems since then.